Just to be fair to the people who come in.
It's not like
You have class.
yeah, but it's not like in 5 seconds they'll miss something.
What do you mean?
They would settle down earlier if you had started.
All right. Gonna get going. So today we're going to get into, as we talked about before, the actual tree of life and how we study sort of the early events in evolutionary history. As a reminder, again, previous lecture we talked about inferring phylogenetic trees. We're going to finish up on one little aspect of that at the beginning of this lecture. And then for the rest of this lecture and the next lecture, we're going to talk about, again, the early events in evolution and the structure of the whole tree of life and the major lineages of organisms and some of their features. Just as a reminder, we're going to have an in-class quiz using the clickers for the discussion section tonight. It's going to be about this reading. It's pretty reasonably short for chapter 22. There's just one page on testing phylogenetic methods. It's in chapter 22.2, and then there's one section there where the header is something like testing the accuracy of phylogenetic methods. And then chapter 1.1 and 1.2, which are the introduction to the book, and have some things that we really need all of you to know the big picture on. So there are going to be questions about that too. Yeah?
You said we could have a page of notes for the quiz?
You can bring a page of handwritten notes on a single piece of paper for the quiz. If you like to write small, I mean, the point here is to get you to read and think about the material. And if you do that by transcribing it down onto a piece of paper, that's great. Just no Xeroxing anything and bringing that in. You actually have to do it yourself. Yeah?
How long is the length of the quiz?
The quiz is going to be very short. It's going to be basically the equivalent to 3 clicker questions. So it'll take up the first 5-10 minutes of the lecture or something to that effect. Any other questions about this? Just remember, bring your clickers. And if you want the details, the way it's going to work is there will be 6 of these in-class quizzes. Each one is worth 10 points, and you will be able to drop the lowest of your scores so that we count 5 of them in total for each person. Yeah?
How many questions out of?
It's going to vary depending on the test. So for today's, there are 42. No, they're going to be 3 today. And this is like a warm-up exercise, so don't have too much fear of today's. Any other? Yeah.
So are quizzes end up being in the Monday discussion section, or are you going to alternate them on Wednesdays or something too?
The original plan is to alter them. It's just going to depend on the timing of the class and where we are relative to when we sort of have planned for certain quizzes. So it's a little bit unpredictable based upon how we progress. And we'll try every time to give you, one lecture's worth of warning that they're not meant to be surprise quizzes. They're meant to actually get you to do something. So surprise is not our goal here. All right. And then the reading for this week for part of the Monday lecture this evening and Wednesday and Friday is chapter 26. So we're going to do this wrap-up of the one thing I got to at the end of the last lecture, which relates to phylogenetic methods, in particular, well, 2 things really. One relates to assumptions that are made about the parsimony reconstruction methods, and I started going into this discussion of counting different types of changes differently, and I'm going to finish that conversation, and then we're going to talk about how you might deal with the fact that evolution is not incredibly simplistic, and we might want to count different types of changes differently, and that's to talk about likelihood methods, which is another class of phylogenetic reconstruction method. And then we'll switch over into talking about the tree of life. So in the last lecture, I talked about this briefly. There are the parsimony analysis that we did in the class is sort of really important for giving you the general principles for how phylogenetic reconstruction works. But there are many, many, many assumptions that we made in the course of doing it the way we did it. And I listed 3 of these assumptions here as just examples, that all types of characters are treated equally, that all types of changes are treated equally, and we're going to talk about this, again, I started in on this but didn't finish this in terms of DNA, types of DNA changes, and then changes on all branches are treated equally, and I will very briefly talk about this. And in general, these assumptions are fine for some purposes, but in most cases, they're oversimplifications. And if you can, it's important to try and account for the reality of evolution when you try and make inferences about evolutionary history. So I started in on this discussion of the different types of DNA changes that occur when the sequence of DNA changes between one generation and the next. That's known as a mutation, when the genome of an organism changes. And one way that the genome can change is just the letter, the base at a particular site can change to one of the other bases. So there are 4 DNA bases. If you have any particular DNA base at a site, it can either stay the same or change to one of the other 3 bases. And as I mentioned, the DNA bases come in 2 chemical forms, the purines, G and A, and the pyrimidines, C and T. And when the DNA forms its normal double helical structure with 2 strands of DNA pairing with each other, you always have a purine pairing with a pyrimidine or a pyrimidine pairing with a purine. And that basically keeps the width of the DNA double helix constant. You have one of these slightly larger bases paired with one of the slightly smaller bases. And so you always have this purine-pyrimidine pairing. When you have changes from one generation to the next in the sequence of a particular region of the DNA, if the change is from one purine to another purine, that is A to G or G to A, or from one pyrimidine to another pyrimidine, that is C to T or T to C, those are known as transitions or base transitions. If you have a change from purine to pyrimidine or pyrimidine to purine, those are known as transversions or base transversions. And the reason we're talking about this is that when people have actually looked at the changes that occur in organisms, either in the laboratory where you can take a single cell and grow it over many generations and look at what's different at generation 100 from generation 1, you can look at the mutations that arise, or you can do this by comparing the sequences of different individuals in a population or different individuals from different species. And when people do this, they have discovered that in general, in most of organisms and across most of the genome, the probability that you get a change, it's much more likely that you would observe a transition change than a transversion change. So again, DNA transitions are generally more common than DNA transversions. Everybody there on this? Okay, so the reason I'm telling you this is when you realize that the changes are of different frequency, you might want to take that into account when we do parsimony reconstruction. So we were treating all changes equally. Whatever they were, we counted steps. And every change got treated equally. And you might want to not do this. And I'll give you sort of the example not to do with phylogenetic analysis. Again, everybody uses parsimony type analysis in their daily lives. You're walking down the street, in the middle of Davis, and you see a person lying on the side of the road with their bike in a pile and, blood on their head, and they're crying. The assumption, probably pretty common in the first few weeks at Davis. The assumption is going to be probably that there was a bike wreck. Now, it's possible that someone actually picked up a bike and threw it at this poor person. Or threw it off a building at this person, or something to that effect. Now, but what you're going to do is in your head, you have this sort of probability matrix for what's a common event and what's a rare event. And you're going to use parsimony to say the most probable thing I think occurred is the thing that is more likely, more common. And that's what we want to do with phylogenetic reconstruction. We want to penalize changes if they are considered rare or unlikely. If we have a tree that requires a transversion event, and we're comparing it to a tree that requires a transition event, and that's all the differences between the two trees are, the best tree is probably the one that requires a transition event because transitions are more common. So the tree that has this transition event occurring somewhere in the tree is going to be the more parsimonious tree, just like you're going to infer that the person had a bike wreck instead of someone threw the bike at them. So that's what we're going to apply, that same general principle to our phylogenetic reconstruction. So here's the reconstruction data that we had from before with 3 informative characters, 3, 6, and 7. It turns out that these characters came in 2 different flavors in terms of transitions and transversions. I'm sorry, the color here, as people pointed out in the previous section, the colors don't match. So transversions are in red up here. Transversions are in blue in these columns. So I'm going to walk you through this. Character 3. There are 2 character states, G and A. The changes that we inferred occurring along the tree were either A to G or G to A changes. Those are changes between purines, and therefore those are transitional changes. C to T, for character 6, the changes that we inferred on the trees for character 6 are also transition changes. They're changes between pyrimides. However, character 7 is sort of like the throwing the bike at someone example. These are transversion differences. C to A, C is a pyrimidine, A is a purine. We would have to infer the occurrence of transversion changes for this character. So when we scored everything equally, the tree that won was tree number 2, right? It had the lowest total score. But perhaps we don't want to do that because of this difference in the probability of transversions versus transitions. The way we account for that difference is to score the rarer events with a higher number of points, basically. The rarer the event is, the more points we're going to give it, and that way it counts against any tree that has those events. So let's imagine maybe we want to count transversions twice as much as transitions. If we did that, we would have 2 points here and 4 for each of the other 2 trees, as opposed to 1 and 2. What this is doing basically is that tree number 1 required 1 transversion event. Tree number 2 and tree number 3 required 2 transversion events. We are going to penalize tree number 2 and tree number 3 extra because they required 2 instead of 1 transversion event. So we have doubled the cost in essence. Instead of the difference between tree 1 and tree 2 being 1 point, the difference is now 2 points. And that means now that tree 1 is tied with tree 2. So by penalizing transversions, we have changed which tree is the winning tree. Now we have a tie instead of tree number 2 being the winning tree. We can make it even more. We can say transversions cost 3 times what transitions cost. If we did that, now tree number 1, again, which only required 1 transversion, is penalized a lot less than the trees that required 2 transversions, and tree number 1 is now the winning tree. So this weighting of different types of changes can have profound impacts on our phylogenetic conclusions. Does that basically make sense here? Just again, like the bike accident example. You need to take into account the probability of events if you want to do this correctly. Okay. So here's a clicker question for everyone. What if we had transversion events counting 5 times transition events? which tree is now going to be the winner here? The answers are over here. A for tree 1, B for tree 2, C for tree 3, a D for a tie. So again, what if transversions counted 5 times transitions? And you can talk to your neighbors again. You won't be allowed to chat with your neighbors on the quiz tonight, but for clicker questions, you are allowed. What? You don't need them, based upon what we just went through. Another 10 seconds. Three, 2, send in your answers. All right, good. So most people got it correct. The answer is A. And if we go back, remember when we counted it as 3 times as many, basically tree 1 was now the winning tree. And we've converted from tree 2 being the winning tree to tree 1. If we now make the scores be 5 times instead of 3 times, this would be a 5, and these would both be 10. And tree 1 now is even more better, more excellent than the other 2 trees. So you should just have a general understanding here that the weighting is basically shifting which tree is the winning tree depending on the number of the types of rare changes that occur on each tree. If a tree has a lot of rare changes on it, the more you penalize rare changes, the worse it's going to do. If a tree has fewer rare changes, the more you penalize rare changes, the better it's going to do. So now I made up these numbers, 1x, 2x, 3x, 5x. In practice, making things up for phylogenetic reconstruction is not the smartest approach in the world. And so what people try to do is take real data and determine what the best ratio of these different types of changes is. So you can compare rates of transitions and transversions in different data sets and ask, what is the difference? Maybe you should weight it just by the difference in the rates. You could take the data, like in the example that you're going to be quizzed on later tonight, for testing phylogenetic methods, and use that data where you know the answer, and then see what weighting is the best way to recapitulate the truth in a phylogeny. So there are many different ways to go about trying to figure out what the best ratio is to use for this weighting, the best weighting number. And a lot of phylogenetic analysis involves trying to figure out, in essence, what the patterns of evolution are for different characters so that we can account for their variability between and within taxa. Does that make sense, what we did here? Any questions about this? So you can imagine basically a lot of other approaches where the default assumption where we treated sort of everything in the tree equally might be violated. And another one is that we were treating basically changes on all branches equally. And I'm not going to go into the details of how you would correct for differences in different branches, but I want to just go through a little example of this and how we represent this on phylogenetic trees, because you will see a lot of phylogenetic trees that are trying to show that the rates of evolution in different lineages on different branches differ. Again, we're not going to talk about how you would correct for this in phylogenetic reconstruction methods, but suffice it to say that you should try if you knew how. So an example of this involves the HIV virus. So the HIV virus makes an enormous number of mistakes when it copies its own genome to generate new HIV viruses. The rate at which it makes mistakes, that's known as the mutation rate, is extraordinarily high. Compared to humans, it's 6 or 7 orders of magnitude higher. I mean, it occurs at, million-fold higher rate than in humans. When we copy our own genome to produce offspring, either for sexual reproduction or just our cells copying our own cells, the rate of mutation is actually pretty low. Humans and many other organisms have detailed mechanisms to try and prevent errors from occurring in the first place, and then if they occur, to correct them. HIV, the virus, has basically gotten rid of all of these mechanisms. It, in essence, purposefully makes mistakes when it's copying its own genome. And this leads HIV to be really annoying in terms of treating infections with HIV. So in the course of one infection in an individual, within a few rounds of HIV replication, you can get billions of different viruses, each with a different sequence from each other. If you treat someone with drugs when they're infected with HIV, the probability is very high that one of those variants that arises is going to be resistant to that drug. It's one of the reasons why HIV is treated with 3 drugs frequently to try and prevent the virus from escaping by mutation. Yeah?
It evolves so fast. How does it stay so effective?
Yeah, that's a great question. People don't necessarily know the exact answer to this, but basically what HIV is doing is massively sampling the possible genetic diversity that is related to the original virus. So it makes lots and lots of mutations, makes massive population numbers. But then there's a very strong selective window, so that it may be making lots of variants, but the bad ones don't make it through to the next generation and infect another cell or transmit to another offspring. So the selective pressure is enormous, and most of those variants are dead. But because it has this massive population size, it is able to evolve resistance to various drugs and to the immune system very rapidly. And this is damaging. So many organisms that have very high mutation rates, like HIV, they have problems that come from having that high mutation rate. But to HIV, it's generally a benefit, and it has sort of maintained this high mutation rate. So if you compare 2 organisms, let's say you start with a common ancestor, split into 2 lineages, and then one of those lineages throws away all of the error correction mechanisms for protecting its genome, its evolutionary rate will go up. And the way we represent this in a phylogenetic tree is by having its branch length be shown as longer. So on the left here, we have the normal phylogenetic tree that we've been talking about so far in the class. That's called a cladogram, when you're just showing the relationships among organisms in the tree. The branch length in this tree does not mean anything. The tree on the right, the branch length is trying to represent the amount of change that has happened on that branch. In essence, the longer the branch, the higher the rate of change. And that's called a phylogram. And in many of the phylogenetic trees, you will see, both in this class and in any type of evolutionary study, it's very common for people to use these phylograms instead of cladograms. So you should understand the meaning of them. The longer branch basically means a higher rate of evolution compared to its sister taxa here. It still shows the branching order, so the monophyletic groups are the same between these 2 trees. It's just in some of the branches, the rate of evolution has gone up or gone down, so we're showing differences in the length of the branches. So just like with different probabilities of different changes, like transitions and transversions, if you were building an evolutionary tree of organisms and some of them evolved really rapidly, like HIV, and some of them evolved really slowly, you might want to count any changes that occur in the lineage that evolves rapidly in sort of the same way that we were counting transitions. If an organism has a very high mutation rate like HIV, changes in its branch are to be expected. they are not to be penalized against, whereas changes in the human lineage are less likely, and therefore maybe we should penalize them. So phylogenetic reconstruction methods also try and account for this difference in the rates of evolution in different lineages. Now, when you're doing parsimony character state reconstruction, this can start to get pretty messy, and the parsimony methods are not particularly adept at dealing with all of the differences that can occur in patterns of evolution between taxa. And that's where these likelihood methods come in. Now, we're not going to teach you all about likelihood methods. I'm going to spend one slide on likelihood methods. I would love to talk to people more about this if they want to, because this is what many people use in practice now when we're doing phylogenetic analysis. So the way likelihood methods work is very similar to parsimony methods. You take your taxa, you identify the possible trees that could relate those taxa, and you give the trees a score. And you pick the winning tree based upon the tree having the best score. And you go character by character, adding up the score for each tree, and then summing up across all characters for each tree and pick the winning tree, just like parsimony methods. The difference is how we give it a score. What likelihood methods do is much more complicated than what parsimony methods do. So likelihood methods start out with a model. The model is a sort of conceptual mathematical description of how characters change between states. And this model allows you to represent things like transitions being much more likely than transversions. So you have a substitution probability between A and G, and between A and T, and between A and C, and a substitution probability between T and A, and so on. And you can represent the probability of all sorts of different types of changes in this model. And then what you do is you take the model and you ask, given tree number 1, and given my model of how sequences change, what is the probability that I could get the data that I actually observe? And then you do it for tree number 2. Given the same model, but now tree number 2, what is the probability that I could get the data that I observe? And one of the reasons that this is really powerful is it allows you to really easily account for different rates of evolution in different branches. If a branch is longer, the probability just goes up that you can generate certain types of changes on that branch. You can also account for different rates for different characters really well. So I mentioned these 3 assumptions. Characters might be treated differently. Types of changes might be treated differently. Branches might be treated differently. Likelihood methods allow you to deal with these variances much more accurately. They also happen to be computationally much more expensive, much harder. And so they crunch our computers much more. And for a while, they weren't used that much because of this computational limitation. With fast, my iPhone can do better phylogenetic reconstructions than any of our desktop computers could do 4 years ago. So likelihood methods and similar methods are sort of taking off now because they allow you to account for these types of patterns. We're sticking with parsimony methods in the class because the principle is similar. Mathematically, they're much easier to describe, and they allow us to discuss character evolution in many ways that is very useful for the rest of the class. But you should know that likelihood methods tend to be the things that people now use in practice when we're building phylogenetic trees. Are there questions about likelihood methods before we move on? There's a third class of methods that we will get to either at the end of today or probably the beginning of the next lecture later tonight. And they're called distance methods, but we'll come back to that. So now we're going to shift for the last 20 minutes or so here to talking about the tree of life itself, rather than just theoretical concepts and how you infer things. What does the structure of the tree of life actually look like? And the first part in this sort of exercise is to ask a very simple question. How many trees are there? That is, another way of thinking about this is, how many times did life originate on this planet? Are there two separate groups of organisms that each have a separate ancestry from each other? Are there three? Or maybe does all life relate to a single common ancestry, and therefore, in essence, there was a single origin of life? And the way we're going to answer this is through these things called universal homologies. And that's what I'm going to walk you through. Oh, just as an aside, I don't know how many people have been in the Oak Grove and seen the really nice Tree of Life sort of sculpture there. It's on the outhouse. I don't know if that's like a message to me or something. But there's this really gorgeous Tree of Life sculpture on the outhouse over in the Oak Grove. Okay, so we've been talking in this class, and the rest of this class will be about the remarkable diversity of life on this planet. Diversity of shape, diversity of behavior, diversity of physiology, diversity of biochemistry, diversity of molecular biology, incredible differences among organisms. Yet, despite these differences, there are things that all organisms share in common with each other. And that's part of why I want you to read chapter 1.1 and 1.2 for this class, because it discusses some of those things that everything shares in common. And so, for example, one aspect of all organisms that we have in common is the general processes of information inside cells. So something called the central dogma, which is that DNA is the genetic material. It is transcribed into RNA, which is in essence a message, and that RNA is translated into protein. All organisms on the planet do this. So that central dogma is found across all organisms. The mechanism by which RNA is translated into protein via a 3-letter genetic code is found in all organisms. All organisms use this 3-letter code. All organisms use the same machines to carry out this translation. The exact nature of the code can sometimes vary, so the which letter codes for which amino acid has a little bit of variance between taxa, but everybody uses a 3-letter code, and everybody uses these ribosome machines that are very similar to each other to carry out this process. The structure of the ribosome machine is basically identical between all taxa. This is a 3-dimensional crystal structure of the ribosome, and if you compare that 3-dimensional structure, it includes something like 40 proteins, something like 3 to 4 to 5 different RNA molecules, as well as sometimes a few other modifications like methyl groups and things like that. And the way it folds together is basically identical between all taxa. And it's the machine that carries out the translation of RNA into protein. If we compare molecular and cellular features like this, as well as sort of specific cellular complexes and genes, we find a large number of what could be called universal characters, like the use of DNA as a genetic material, the use of A, C, T, and G bases in DNA, and A, U, C, G bases in RNA, the 3-letter code, as I mentioned, the central dogma, as I mentioned, the fact that all cells are surrounded by a lipoprotein cell envelope, that all cells utilize 20 core amino acids in their proteins, and even if you look in more detail, the actual structures of some of these cellular complexes, like the machine that carries out translation, are basically identical, universal and identical between taxa. So you can look at this list across all organisms, and there's one final thing you have to do in order for this list to be meaningful to studying the origin of life. And that is to make either to figure out or to assume that these things are homologous between all taxa. So remember, similarities between taxa can arise by 2 main processes, or in 2 main ways. Common ancestry, similarity due to homology, or convergent evolution. So what you have to do is ask, are these features indicative of homology, or are they indicative of convergent evolution? And it's actually not the simplest exercise to figure out in many cases, but what people basically ask is the following. If life evolved separately, what is the probability that it would use DNA as the genetic material? What is the probability that it would use these 4 particular bases, or these 4 particular bases in RNA? What is the probability that it would use a 3-letter code, that it would do things in this way, that it would have 20 core amino acids, and so on? And the conclusion is that the probability that all of those things would happen in exactly the same way in different lineages, even with very strong selection, is very low. And that these are not just universal features, but they are universal homologies between all organisms on the planet. So again, characters found in the same state in all organisms can be called universal. Characters inherited from a common ancestor, as we've already talked about, are called homologous. So homologous characters that are universal, these are these universal homologies. And it's the universal homologies that tell us, basically, that all life has a common ancestry. And so I'm going to show you this in a phylogenetic form. So here's a phylogenetic tree, that one we've been using throughout the class. Here's the most recent common ancestor of all of these organisms. If these were all the organisms on the planet, and this is what the tree looks like, that's basically saying that life evolved once. They all share a common ancestry with each other. But the tree could look like this. You could have one set of organisms that share a common ancestor with each other, and another set that share a separate common ancestor, and these lineages are not connected to each other. And that's in essence saying life originated twice. So these are, the pie could have had 3 times or 4 times, etc. In terms of features, if we have a feature present in all of these organisms, A, B, C, D, E, F, G, H, and if we have this tree over here, we are saying that that feature evolved once. And if we have this set of trees over here, we're saying that feature had to have evolved twice. There's no way that these lineages get connected. So again, if we are saying a feature, like the ribosome, is homologous between taxa, it means that this tree is impossible. The features we are explicitly saying only evolved once. So we have to have a tree where all organisms share a common ancestry. That's what universal homologies tell us. That there was a single origin of the ribosome sometime prior to the existence of the most recent common ancestor of everything. There was a single origin of DNA as the genetic material sometime prior to the existence of the common ancestor of everything. So the universal homologies tell us that all life came from a single origin. Does that make sense? So one key thing here is that here's one phylogenetic tree diagram, here's another. The structure of the tree makes no difference to this inference. The universal homologies tell us that all life had a common ancestry, but it doesn't matter if humans are sister taxa to chimps or if humans are sister taxa to dogs. That is of no importance to figuring out that these universal homologies tell us that life had a common ancestry. So we don't need to know anything about the structure of the tree to make this inference. That's a separate activity. So again, the existence of universal homologies implies a single origin of life. So now we're going to get into the actual structure of the tree of life itself. Now that we know that it's basically one tree, we have to figure out how things are related to each other on that tree. So another clicker question here. True or false? The presence of universal homologies can be used to infer the phylogenetic relationships among organisms. All right, everyone ready here? Couple seconds. All right. All right. Maybe that people are overthinking this question a little bit, but we can talk about that. So for the people who answered, so the answer I would like it to be is B, false. So I'm curious though, because we can edit the system. If people can explain to me a reason why they put true, I'm happy to give credit for this to people. So who put true? And can you explain it? Yeah.
I put true because it was like the example where you were showing two different trees. So it shows that all the lineages stem from one common ancestor rather than two different trees.
Right, but that doesn't tell us what the relationships are among organisms. It's every taxa has present for the universal homology.
I just thought I could show it that they were all connected.
You can show they're all connected, but we can't show what order. So the phylogenetic relationships that I meant here is what is the order. That's what phylogenetic relationships means. So we show that they're all on one tree, but we have no idea of how they fit together on that tree from this information. Anybody else? Yeah.
I was thinking if we could say that everything started with like looking like this or with these traits, then obviously all the changes didn't occur at once. We didn't just all of a sudden appear like this. We can use that starting position to show each individual change occurring from there.
Yes, although in this tree, so the problem is that all of those changes occurred prior to the existence of the most recent common ancestor. So figuring out the order in which anything occurred doesn't help us tell how organisms are related to each other, right? Because they all occurred back here on the branch leading up to the common ancestor, because the common ancestor has all of these features. Does that make sense? So it's interesting to figure out what that order is, but that relates to the origin of life, really, and not to the relationships among organisms. Yeah?
It could show how it's related to the tree that has all the organisms, because if it's not universally homologous, then it would belong to a separate tree, and you wouldn't know anything about that organism.
Yeah, I mean, if an organism did not have one of these features, it wouldn't be on our tree. But they're universal feature. They're universal homologies, right? Every organism has them.
Yeah, so say you discovered a new organism, and it did not have that.
Yeah, you're stretching it, but yeah, I can see how you might say that the absence, so not the presence, right? The absence of universal homologies might allow you to group together organisms that don't have it, but we'd have to sit down and talk about this. I'm not completely convinced that. So there is one interpretation that I haven't heard from anybody yet for why this is a poorly worded question. And that relates to what we're going to talk about next, which is that it turns out that universal homologies are in fact present in all organisms, but they're not identical in terms of their underlying components in all organisms. And you can use the differences inside of these features to distinguish phylogenetic relationships. Much like the presence of a backbone can't tell us relationships among vertebrates, because all vertebrates have a backbone. But the exact shape of the backbone, the exact position of some of the different ridges and things like that can allow us to study the evolution of different organisms. Inside universal homologies, there are these types of features. I'm just going to quickly go through this just to sort of beat the point into the ground before we get into this inside universal homologies. So this is what the data matrix looks like for universal homologies. The presence of universal homologies. That's what I meant by the question. There is no information here that is phylogenetically informative. You can't infer the relationships among organisms from this information. It's like the invariant features from our data matrix. And what we need is variation to do this. Same as for any other evolutionary reconstruction. We need features that vary between taxa in certain ways in order to infer their relationships. And people have tried to do this type of activity for many years. They have tried to build a tree of life by comparing features among different organisms and then trying to infer the branching order. And in most cases, when people did this, they would infer the branching order among, say, plants and the branching order among animals. And then they would shove plants and animals together in a guess, basically. And then they would say, I have no idea what to do with the small things, the microbes. So there's a classic evolutionary tree by Ernst Haeckel showing 3 major lineages of organisms: animals, Animalia, Plantae, and Protista. And then bacteria are somewhere down at the bottom of the tree and with no detail, because he had no clue what to do with them. A more recent tree that many people refer to as the 5 Kingdom tree by Whitaker, with 5 major groups of organisms. And again, the bacteria which are lumped together into the Monera here were basically not dealt with well. And the reason for this was the inability to make sense out of these universal homologies. That is, everything has them. That's all we can say, so we know everything's on one tree. They put everything on one tree. But they didn't know how to relate everything to each other. And that changed basically with this person. So Carl Woese, who's about 90 years old now, I think, or so, at University of Illinois, was studying the mechanisms of translation. He was looking at ribosomes in different organisms, and he was very interested in sort of the molecular details of this. And one of the things that he did was he took out the parts of the ribosome from different organisms, pulled out the RNA molecules from them. So ribosomes are actually made up of protein and RNA. The RNA is called ribosomal RNA. And he pulled out that ribosomal RNA, and he ran chemical reactions on that RNA that allowed him to determine the string of letters inside that RNA. This is now generally called sequencing. It was very painstaking when he and the people in his lab did this. It's much easier now. It took him, years to do this for one species. And now we do millions in half an hour. But it was incredibly painstaking. And what he did was he read the string of letters in different taxa of these RNA molecules inside the ribosome. And it turns out that even though the big structure of the ribosome is basically the same between all taxa, the string of letters that different organisms use inside the RNA molecule, inside the ribosome, varies. It doesn't vary a lot, but it varies. And what he did was he took that data and turned it into a data matrix, basically. And he built an evolutionary tree from one of these data matrices. We'll come back to this later tonight. And I just want to show you what that tree looked like. So this is the tree that he got from doing this from his data. And what was particularly surprising about this tree, well, first, it was the first tree that actually used data to link together all organisms on the planet. And second, and surprisingly, the organisms that people had called prokaryotes, that is, the organisms that did not have a nucleus packaging their DNA, were actually composed of 2 completely distinct groups of organisms. The bacteria, this is what they're now known as, and the archaea. He originally called them the archaebacteria, but they're now known as the archaea. And that's what we'll talk about in the second lecture tonight.